right, it looks like we're not getting any more new people coming in, so we're going to go ahead and get started. Thank you all for coming. I'm Bree Price. I'm the WSUB Program Extension Coordinator. This is our first webinar of 2026. We have three more planned for this year. If you're interested in what those are about and to register, please visit our upcoming events page on our website, bees.wsu.edu. I just have a couple announcements before we begin, and then I'll introduce today's speaker. The WSU Honeybees and Pollinators program is a cornerstone of the College of Agricultural, Human, and Natural Resource Sciences, abbreviated CONORS, that is dedicated to fostering resilient ecosystems in Washington and beyond. Our mission intertwines innovative research, community engagement, and education to safeguard pollinators that are pivotal to our food security and environmental health. In partnership with the Connors Resilient Washington Initiative, we're committed to advancing sustainable practices and pollinator-friendly landscapes, as well as ensuring a flourishing future for agriculture and natural resources. There will be time to answer questions after the presentation today, so feel free to type questions into the Q&A box below anytime during the presentation. After the event and before you close your browser, you should be prompted to answer a short 5-question education and outreach survey. Your participation in this will really help us understand our impact today. Today's speaker is Dr. Ryan Kuesel. Ryan is a postdoctoral researcher in our WSUB program. Ryan is originally from Holland, Michigan, and he earned his PhD at the University of Kentucky in the Gonthier Lab studying berry pest management. At WSU, Ryan has partnered with commercial apiaries to inspect their hives for signs of disease and to monitor their growth. He's also educated current and future veterinarians about honeybee diseases. Ryan's current major project aims to better understand European Foulbrood. In this webinar, Ryan will outline whether we found differences in European Foulbrood infections or honeybee colony strength in commercial colonies that were or were not in blueberry pollination. This data is one part of a multi-state project funded by an SCRI grant where migratory honeybee colonies were tracked as they were moved between crops, almonds, apples, blueberries, brambles, sunflowers, and canola in 2023 and 2024. Today, you're going to get a first glance at this project's results. I'm going to go ahead and hide my screen and you can share, or hide myself and you can share your screen, Ryan.
Cool. Yeah, thanks so much for the great introduction, Bree. And thanks everybody for coming today. We'll share screen 2. Actually, PowerPoint slide. All right, how does that look? I think we're good.
Looks good.
Perfect, thanks. All right, yep. So, um, like Bree said, thanks so much for the introduction. Looking at the incidence and colony impacts of EFB in the Pacific Northwest based on that 4-state study that we've done 2 years out of 5 years so far. So a quick overview of this webinar. I'll cover what European Foulbrood is as a bacterial species and what its symptoms look like in a colony first to get us all on the same page about what European Foulbrood is. Then also go into how it causes damage inside of bee larvae, and then go into why some beekeepers and researchers talk about it in association with blueberry pollination. Then I'll give an overview of what we know from what we've currently done in the 2024 to 2027 four university study that I collaborated on. Our fieldwork is done, but the laboratory work is just getting started and moving forward. And then I'll give an update on the results of the fieldwork that we performed so far, at least what WSU had collected in the field data-wise, before giving a conclusion about what this all means and then what's to come, which y'all should look forward to in the future out of this project. All right, let's dive into European Foulbrood. You probably all know that European Foulbrood is caused by a bacteria. That's Melissococcus plutoneus. There's a second Foulbrood called American Foulbrood, which is devastating to a colony, much more so than European Foulbrood. But this work, again, is about European Foulbrood and not about American Foulbrood. European Foulbrood is shortened, often abbreviated to EFB as the acronym. So you'll hear me say EFB a lot. It's a lot fewer syllables to say throughout the webinar, meaning European Foulbrood. So EFB infects larvae at a young stage and kills them before they're capped off by nurse bees. So unlike American foulbrood, which kills the larva after they're capped off, you're going to see your symptoms and your larvae die when they're still larvae as they're growing before the capping happens by those nurse bees. EFB is quite a weird, sort of enigmatic disease because it seems to come and go with the passing of seasons. EFB symptoms are much worse in late spring and early summer, and especially in the Pacific Northwest, it seems almost ubiquitous that you'll see a little bit of EFB or a lot. Hopefully not, but you may see a lot in your colonies. Publications as far back as about 1961 mentioned that sudden outbreaks of EFB are followed by a spontaneous recovery a few weeks later, meaning that you may get a sick colony that recovers on its own with or without treatment, which is antibiotics. One specific type of antibiotics for this, oxytetracycline. And as we're learning more about EFB in the modern day, we're slowly finding patterns as to when and maybe why EFB symptoms emerge in colonies. One paper out of 2010 taught us that the outbreak of EFB disease seems to be linked to colony stressors, especially or including a lack of good quality food. So that may be one key to EFB. And recent research has shown that bacteria that causes EFB, Melissococcus plutoneus, is sometimes present in colonies regardless of whether they're displaying symptoms or not. There's a study out of 2024 by Mallory and colleagues at the University of Guelph in Canada who looked at EFB in colonies and said that although the presence of that Melissococcus plutoneus, EFB bacteria, is associated with EFB symptoms, it does not consistently predict the manifestation of symptoms, meaning that just because that bacteria is present in a colony, it doesn't mean that that colony will show symptoms. And that's emphasized here in a second study in 2025 when Peter Fowler and colleagues from Michigan State University looked for EFB bacteria by searching for their DNA in migratory colonies. And when they found EFB bacteria in a given colony, only 28% of those colonies with bacteria were showing symptoms at the time that they were sampled, while the remaining 72% of infected colonies had no symptoms of EFB while they were still had that bacteria present. And that was out of almost 300 colonies inspected. I think 284 colonies that they sampled and inspected. So not only is EFB strange and difficult because it comes and goes with the seasons, it's also weird that the bacteria seems to need maybe some sort of additional factor to let them take over a larva and show symptoms in the colony. That's why I'll often talk throughout this webinar in terms of seeing or not seeing EFB symptoms more so than seeing EFB infection, because being infected and seeing those deleterious symptoms is not one and the same with this strange bacterial disease. Here's the causal agent. Again, it's Melissococcus plutoneus. It looks like this on the right, with multiple circle-shaped cells often stuck together in a line as they've divided out outward in a line. Here, there's four different Melissococcus bacteria or cells smashed together in one line here. It's not spore forming as a bacteria, luckily, so it's much easier to kill than the American foul brood, and you can sanitize your equipment much easier for this European foul brood than the American foul brood because it doesn't form spores. It's also microaerophilic or entirely anaerobic, meaning that it can't survive in environments with high levels or any oxygen. So instead, it needs to survive in really oxygen-poor environments like bee bread, honey, or the inside of a larva, of course, or maybe inside of the soil, which just a few studies have started to show. Larvae are infected with that EFB bacteria when they eat royal jelly or bee bread that contains those bacterial cells. And the bacteria begins to colonize that midgut of the larva really rapidly. And as they multiply in the midgut of infected larvae, they compete with that larva for food and end up starving out that larva. So in weaker larvae, the bacteria will take over the midgut completely. The larva will change color from a healthy pearly white. Here you see nice healthy larva on the left, nice white color. And they're even in it looks like very new comb. That's a light color. Those pearly white larvae will give way to an increasingly yellow, almost brown uh, larva, as you can see here in that right image. It's almost like a progress if you go right to left on this middle column where they start out sort of yellowish white and go right once to get to a little bit darker color. And then these larvae actually start to break down. This larva is dead at this point, and the body's just sort of collapsing before they finally turn into this just brown, almost scale that's loosely stuck to the bottom of this cell. You can see typically with this, because of the body discoloration, a net of white trachea. That's the respiratory system in these larvae moving throughout it. But if you have this sort of deflating, they say like sickle-shaped or C-shaped larva, the larva is falling forward out of the cell and then finally deflating, you likely have uh, European foulbrood. And then while we're moving this scale, or even here is a much more dried out scale, um, the mouth parts of nurse bees can become infected with the European foulbrood bacteria, and they can transmit it throughout the colony. So Melissococcus plutoneus weakens the larval gut microbiome, also, and allows secondary infections of bacteria like Achromobacter eurydice, Enterococcus fecalis, and Pennibacillus alveoli. And it's thought that one or multiple of these contributes to a bad smell that's sometimes found with EFB cases. Then the death of some larvae, but survival of others leads to what's known as a shotgun brood pattern. You can see that clearly here. Some of these larvae have died out, while others are in their final stage before getting capped, and some of them have survived up until the capping phase. That's a common symptom of a lot of different diseases. So you'll have to also look for the state of those larvae. Look for those uh, um, drying up yellowish brown larvae as well. And again, it may or may not have a foul odor if it has an association with those other secondary infectors. The royal jelly might also be discolored. If you can see deep down in that cell with a good light, the royal jelly is yellowish or almost brownish. So that royal jelly that was fed to this larva by a nurse bee is infected with that foul brood, and it's going to get infected by eating that royal jelly. So, of course, the way that EFB gets into the larva is through feeding. So, real quick, when royal jelly or bee bread enters the mouth, it first moves through the pharynx and into the esophagus, which are just muscular tubes that assist in swallowing food, which moves it deeper into the body and into the mid-intestine, which is a huge pouch, major part of the gut that's about a third of the thickness of each larva. So, most of a larva is this mid-gut. Uh, and that structure digests the food and diffuses nutrients across its wall into the hemolymph, which is the blood of insects, and then that's how the larva gets the nutrients that it needs out of that food. And then food is moved into the hind intestine, where water, salts, and amino acids are removed, and then feces is formed and it's excreted. But the as we know, EFB infects this larva in the midgut. So it's this very large organ that is responsible for capturing nutrients that is getting infected by EFB. EFB bacteria start growing between the food mass in the hollow middle of the midgut. You can see an A. The L is lumen. That's the center of this midgut. There's probably food in here, but what we see stained is these red European foulbrood cells, and then purple larval gut cells there. The bacteria is directly attaching to those epithelial cells of the gut on the inner wall of that gut. Uh, and the EFB cells will use up the nutrients that would normally feed the larva's own growth by blocking absorption of nutrients across that purple gut membrane. So as they're stealing nutrients, they're reproducing and they're colonizing all of the gut, as you can see here in C. Now this entire center of the gut, the midgut, is infested with European foulbrood Melissococcus bacteria. And while they're colonizing that gut, they're also producing enzymes that dissolve the gut wall of that larva. Now, if the larva is strong and well fed, it might survive through until pupation, even while infected. They can live to adulthood, but typically those larvae that were infected will come out as slightly smaller, sort of underfed adult bees. But if the larvae are very weak and not well fed, then the EFB succeeds at fully starving that larva, and the larva will die out before the cell wall is capped. That means that EFB usually kills its host larva, and it succeeds at invading the tissue outside the gut after its death. So once the larva is dead, the bacteria and other secondary invading bacteria use what's left of the body and continue to discolor and dry out that cell. And then they're waiting for a nurse bee to pick it up and transfer the bacteria to other food and to other larvae. So if you need to identify EFB in a hive, and this is what we were looking for in our study, remember that EFB kills larvae before the cells are capped. You'll see a yellowish or brownish deflating larva in an open cells, often with that net of white trachea running through their bodies. And you'll sometimes smell a sickly sweet or say almost like sour urine smell, but not always for this infection. And don't mistake it for the American foul brood, though. Uh, the American foul brood, as opposed to European foul brood, is killing the larvae once they're capped off. So you'll see capped cells that are sinking inwards. Right in the center here, these caps are sinking down, they're not popped out. You may see liquid bacterial inoculum, this orange liquid from the AFB emerging from those cells. And then if you do a matchstick test, which is insert a matchstick or any small toothpick through the wall of a cell and sort of stir up the contents of that cell, and you can pull out the matchstick and it'll have a large rope of larval goo if you have American foul brood, much more so than European foul brood. EFB can have a little bit of ropiness like this, but not nearly as much as that American foul brood. All right, so that's we've all learned or maybe been reminded about what causes EFB. Let's switch over to looking at why symptoms and damage in colonies happen. So back at least in 1991, honeybee pathologists Leslie Bailey and Brenda Ball were asking that same question. They found that the number of infected larvae in colonies were highest when the overall population of brood was high. So here they sampled the number of sick larvae in a colony over 2 years. These black circles is year one, and it's peaking up around, looks like mid or late June to early July. And the same is true in year two. It looked like a little better year with less EFB in those colonies. And then up top at the triangles is the overall number of larvae on a different scale. But what's important to note is that they're peaking all at about the same time. So they're suggested that maybe when nectar flow is beginning in that early spring or late June into July, brood production might be taking off, while fewer bees are spending time as nurses, because instead they're being pushed out more quickly into foraging roles. And that decreases the amount of brood food that each larva is fed, because those nurses have to work harder to feed more larvae with fewer nurses, meaning that those larvae are more likely to starve out and die to EFB. Then further, there's also fewer bees, nurse bees, or undertaker bees to take those dead larvae out of the colony. So more bacterial inoculum is building up in the bodies of these larvae that are sitting in the colony at that time as well. So that altogether, they hypothesized could maybe lead to greater EFB presence in late spring, early summer. So one of the earliest crops that needs bee pollination in the Pacific Northwest is blueberry. Bees often come back from California almonds. They maybe get split into two and then treated for varroa. And then around late April, they're moved into blueberries for pollination. Or they might get moved into apple or cherry orchards, which are also blooming at about the same time. But for a bit now, beekeepers have expressed some concerns that their bees seem to get European foul brood more, or at least express symptoms of EFB more when they pollinate blueberries, and that ends up setting those colonies back for future pollination jobs and summer honey collection. That's a pretty concerning possibility for beekeepers and blueberry growers alike in the Pacific Northwest. It's a huge portion of America's blueberry production is done where we are in the Pacific Northwest. So we started looking to see if there's a link a couple years ago. After brainstorming at the start of our study, we thought of several things that we need to look at as a possible reasons why EFB symptoms could emerge in blueberry pollination. It could be that the nectar flow from blueberries indeed is spurring that high brood production and having more foraging, causing larvae to go hungry and die, and then not be removed as fast as they need to be, like Bailey and Ball suggested. Blueberry pollen also has very low protein and essential amino acid content already. So bees pollinating blueberries might experience that nutritional stress, uh, in that way leading to weaker larvae whose maybe immune systems can't fight off the EFB bacteria. Then fungicides are used at high levels as soon as blueberry flowers drop to protect that new growing blueberries from a lot of different fungal diseases. So bees that aren't removed from blueberry plots fast enough and are there into blueberry formation might be exposed to fungicides, which we're learning now actually can maybe disrupt the larval gut microbes inside of bees, which would interfere with their ability to digest and capture nutrients and could cause those larvae to starve even more quickly in the face of EFB infections. Then that time of year is still pretty cold in the Pacific Northwest, or at least it can be, especially at night. So broods could be developing slower due to that cold weather, giving EFB more time to spread and multiply in each larva. Then maybe different blueberry blossoms or the soil around blueberries might harbor a different or more virulent strain of EFB than other crops. That one would be hard to prove. Or perhaps the high acidity of blueberry pollen could acidify the gut environment of the larva, making it more hospitable to EFB bacteria. Blueberries are acidophiles, they love acid, and their pollen represents that through more acidic pollen. Then compounding all those possibilities is the fact that splits are typically made just before blueberry pollination. And making splits leaves colonies with less pollen and honey stores between the two of them. So for folks who are unfamiliar with splits, making a split is just when you take one colony and split it into two by removing about half the brood, the bees, the pollen, and the nectar stores from the original colony, putting them into a new colony, and then you have two different colonies, and you give this new colony a new queen, or let it requeen itself if you're doing walkaway splits. And we do know already from a previous work, actually by several of the collaborators on this current study, that blueberries provide low quantities of already low quality, like poorly nutritious pollen compared to other crops. This is a study from published in 2019 that was summarizing the work over the previous several years from that. It found that of several early season crops, including meadowfoam, cherry, almond, highbush blueberry, and hybrid carrot, bees collected very little pollen in that highbush blueberry environment. And that was measured using pollen traps that remove the corbicular pollen from bees as they're entering the colony. In blueberries, bees brought just back about 81 grams of pollen per week, compared to meadow foam, or a great pollen source that has about 900 or 693 grams per week per colony. Cherry had about 533 grams per colony per week, and almond had 303 grams per colony per week. That would take place before the blueberry and cherry and meadow foam. Hybrid carrot was the worst of all of them. They don't produce very much pollen. And that led us into our four statewide and four university-wide multi-year project. The team at Oregon State, headed by Ramesh Sagili, was awarded the Specialty Crop Research Initiative in 2024 for a whopping $4.2 million. So thanks to the USDA for funding us on that. And that funding was split amongst multiple professors at four different universities. All these folks are from Oregon State, Washington State, Mississippi State, and then the University of California at Davis. And together, all eight of these professors um, and each of their teams plan to look at several of the possible reasons why those EFB symptoms might emerge in blueberries. The objectives of our massive study were to determine the factors associated with EFB symptom prevalence and intensity, evaluate techniques to manage European foul brood, investigate genetic variation in EFB bacteria in colonies pollinating early season specialty crops, and then develop effective EFB management practices at the very end of all of this work. So if we do indeed find an association with EFB symptoms and blueberry pollination, what we really expected to find and still expect to find is that there's not really one direct reason causing that increase in symptoms. We expect that blueberry pollination maybe comes at a poor time of year with inclement weather that causes that brood chill, slows down brood production, and then a lack of adequate pollen in the blueberry landscape, combined with a lack of optimal foraging weather due to that inclement weather, and too few nurse bees for the amount of larvae that they're producing, might all lead to poor larval nutrition and cause some of those symptoms. And then fungicide exposure, if colonies are left past petal fall and into the fruiting period, would be an additional contributor possibly. So the experiment that we've already completed now in the first 2 years of 2024 and 5 was to move colonies along a typical migratory pollination route, where half of them in May in the second period would go to blueberry pollination, and half of those would go anywhere but blueberries for that period. Each university partnered with several commercial apiaries to get this done, and we moved bees that we owned as well along this path. And we and our partner beekeepers moved colonies from each state out to California for almond pollination in late January, early February. And then we inspected the colonies a couple weeks later, right at peak bloom in February. Once the bloom had all subsided, we moved half of those colonies to blueberry pollination down below in our respective states, while the other half were instead moved to a different um, specialty crop, or most often, especially for WSU, they were moved to a holding yard for that period of time. We inspected those at the end of blueberry bloom, but in both locations, right around May, and then moved them out before fruit set in that blueberry into another holding yard or another specialty crop for pollination in our third inspection period. We inspected that third inspection in July, and then moved those to their final overwintering location, where we inspected them just before winter in October as our fourth inspection. We let all of those colonies overwinter as normal, and then repeated the process again back in late January, early February, by moving them out to California almonds, and then doing the same pattern where those same blueberry colonies were moved to blueberry, and the others were moved to not blueberry. And throughout the 2 years, as colonies died out, we removed them from the experiment and continued on with a smaller cohort so that we were tracking the same colonies for both years across 8 total inspection periods. So here's what our very coordinated first sampling looked like in almonds in 2024. All four university teams worked together on these first colonies out in, I think, around San Joaquin Valley of California. And that was in February 2 years ago, 2024. We learned the ropes of sampling and data collection as a group all together, so we were on the same page. And we opened up each colony at every inspection point and measured the bee population as the frames of bee or the frames that were covered in bees. And we pulled every frame out as well and quantified what percent of each frame was covered in open brood, eggs and larvae, and what percentage was covered in capped brood. So here we'd say maybe this is about 90% coverage, maybe 85% coverage of that capped brood. And there's probably, you'd have to look in a better light, some larvae around here, maybe 5 to 10% of open brood. We do the same on the back, and then do all the frames throughout the colony. We also recorded EFB scores. It was on a scale of 0 to 3. 0 meant that there were no infected larvae in the whole colony. 1 meant there were about 1 to 10 larvae in the whole colony infected. 2 meant 11 to 50 colony or larvae were infected in the colony. And then a score of 3 meant 50 or more larvae in that colony were infected. So that was a good way to say how severely these colonies are infected or not at all. That's only possible if we had um, larvae to look at as well. So at some point you'll see some of these colonies we weren't able to diagnose EFB symptoms in because there simply weren't larvae. We sampled healthy larvae carefully without popping them at all, without damaging their fragile larval bodies. We scooped them out with queen grafting tools and forceps, and then added them to a vial, and then put them directly onto -80 Celsius freezing conditions with dry ice, and stored them for to be analyzed later on for what strains of EFB bacteria are present inside of them. And then we also took samples of nurse bees for multiple reasons. For a head protein count to look at how much protein or vitellogenin is present in the head of these bees, which can directly link to how much brood food they can make and how nutritious of that um, royal jelly brood food that the larva can make, and as well as to dissect them out and look at what bacteria are present inside of their guts as adults. All right, so here's the results so far. This is just data from those inspections that we undertook at WSU. There's more data from the additional universities that we will look at in future talks, and if we look at Oregon State University for those talks and more. So first up, this is just colonies with symptoms. This means the colony had symptoms at any point in any of the 8 inspection periods. We split it into blueberry versus non-blueberry going colonies. And we have symptomatic here at about 30.3% in the blueberry pollination group versus 32.3% were symptomatic for EFB in that non-blueberry group. So it's pretty much exactly even. And then also with the non-symptomatic, they're approximately even as well, about 69.7% in the blueberry group, 67.7% in the non-blueberry group. So already that was kind of surprising that we did not really see a difference at that coarse level for colonies that were ever infected showing EFB, but that's over 2 years of time and 8 inspection periods combined into one. This splits those out among the 8 inspection periods across the 2 years into 5 columns, where green is a 0. There was no EFB located or identified. 1 is yellow, where there's few larvae. 2 is a little more larvae, and 3 has that large infection of 50 plus larvae infected. And then some of those had no larvae for us to identify, so we had to put that in a separate category. Looking first at February, We were surprised to have a couple uh, colonies, about three colonies that we suspected had that EFB infection, but at a pretty low level, about a 1 for two of those colonies, and a 2 for one of those colonies. That shows that EFB can happen outside of that peak season of you know, late spring. In our May survey, this is into that right at the end of blueberry pollination. We're starting to see what we suspected was more EFB in these colonies, with some even reaching that third level. 15 of those colonies had that third level of highly infected with what we suspected was EFB. 34 colonies are at the level of 2, and then 26 were at the level of 1. So EFB seemed to be peaking there. This was late May. Then we skipped June and went into our July survey when they're in that next crop or holding yard. We see the level of EFB is about the same, but starting to go down a little bit, especially in those ones and twos. And the no larva still, both of them have about 24 colonies with no larva. So May and July is our peak. Maybe likely was peaked right in the center of both of these inspection points. In October, we see very few colonies that still had European Foulbrood symptoms. And with some of these, I would be suspicious that it may not be EFB, but may instead be colony collapse of a different variety, like parasitic mite syndrome, where there's too much varroa and you're seeing sort of similar symptoms, but maybe we misdiagnosed those in the field. And then over winter into February. We have very few, again, just 1 and 1. And now we're seeing that cohort really, really decline. Very few colonies survived into the second year. That's shows how difficult beekeeping is, especially when we're not using antibiotics to treat for this disease. In May, we were peaking with our very few colonies still. Just 3, 2, and 1 in that increasing severity category. And then in July, we actually found no symptoms. So it seems like we had a good year. Maybe those bees were able to feed well and survive throughout that year or throughout that summer and beat off the EFB disease. And then in October, again, we see some that we suspected were infected with EFB, but um, maybe a different disorder. Now let's split those into blueberry versus non-blueberry, and we're looking at the percent of colonies within that blueberry group and the percent of colonies within that non-blueberry group. In this is just the first year of inspection points. In February, very few that just show any EFB symptoms at all, so 1, 2, or 3 on that scale. In May, we're peaking at 32.1% of EFB symptoms in blueberries, a little bit elevated compared to our non-blueberries at 22.5% colonies infected in May of 2024. So we have a little bit of differential there, a little under 10% more colonies infected in that first year in the blueberry colonies. Then following that up, as the colonies are feeding off EFB and healing up, we have about 20.5% with EFB symptoms in blueberries and about the same, 20.1% with EFB symptoms in the non-blueberry goers. And then finally into October, we see a little bit more of that EFB might be hanging around if it's not a red herring, um, whereas our non-blueberry going colonies have none. This is into year two, 2025, where we have very few colonies, but are still seeing there might be some EFB at the start. We're having a little bit of EFB symptoms in blueberry and strangely more in our non-blueberry crops this year before moving into July, where it seems to cure itself and heal up. And then we go back into maybe some of those false flags or red herrings in October. It is still possible that's EFB, though. I should stress that. And then this is just in more detail, looking across the three different uh, categories of damage or severity in each treatment. sort of laying that out in more detail. That's for 2024, and there's for 2025 with the severity scores. So really, it seems like so far there's some variation between the two groups. It's very hard to say if there really is a lot more in blueberry, but there may be a little bit, it seems. Now let's look at the fate of symptomatic colonies. This is sort of getting at what's the cost of European foulbrood infections overall. And this is regardless of treatment group. They may or may not have gone to blueberries in this group. And what each of these dots means is you're moving from sampling ground 1, 2, 3, 4 over winter, and then 5, 6, 7, 8, meaning February, May, July, October, February, May, July, October. And green at a 0 is the EFB value or score of 0. That's a healthy colony. Moving up to yellow, hopefully y'all can see it on your screens. The yellow at this level 1 is the level 1 infection. Up to orange is 2, and up to 3 is red. And then down to -1 means it's a black dot. It means that there's no larva to judge this on. And so you can see a lot of these colonies are not making it super far into the season, and you can highlight what all exactly is happening here, where green means that that colony has seemed to outlast the larva or outlast the infection of European foulbrood. Yellow means that it maybe died out due to European foulbrood. It made it 2 sampling points before dying, and there's no points here, meaning that that colony's died out. Uh, Orange up here means that the colony's died out. It had 1 sampling point, and then it died after that, based on when it was infected last. And then in red means that that colony was infected, and then the next time that we sampled it, it was gone. Maybe likely died out to that European foulbrood. So I'd say if we look at this, black means that there were. It's hard to tell because the EFB only occurred in our last sampling point. We don't know the fate of that colony because we stopped tracking it after those 2 years. And 4 of those colonies showed just those symptoms in the very last inspection in October. Green, 12 of these survived more than 2 surveys after the symptoms, which is about half of a year. That's 21% of the colonies that I'd say overcame that infection. Then in yellow, 21 colonies survived exactly 2 surveys after infection. It's about 37% that I say were maybe weakened by European foulbrood and maybe couldn't survive in that weakened state, but maybe an additional stressor took out those weakened colonies. And that stressor very likely might be, especially in these blue highlighted colonies, might be winter. So these colonies made it, 4 periods, and most of them had an infection in that May sampling point 2. They likely either died over winter or maybe were combined by ourselves or by partner beekeepers with other colonies because they were judged to be too weak to survive over that winter. In orange, those survived just that 1 survey after infection and then died. It's about 18% that I say were likely weakened by European foulbrood and likely died as a result of that disease. And then in red, 10 of them died immediately after their infection was diagnosed. So that means quite likely in my eyes these colonies died out to European foulbrood. All right, next, let's take a look at some of those metrics. And this is the frame of bees between the 2 treatments. For all these graphs from here on out, red means that they did not go to blueberry, and blue means that they did go to blueberry. And we have our 8 inspection periods on the x-axis, and then the frames of bees, the number of frames covered in bees on the y-axis up here. So as we're going along throughout the season, seems like the no blueberry colonies fared a little, or started out a little bit stronger, maybe by about a frame of bees here. But in the May survey in blueberries or non-blueberries, they came back to about the exact same before it seems like about 2.8 frames difference occurred in those colonies that did not go to blueberry versus those that did go to blueberry. And again, this is usually these colonies and blueberries were moved to a different location for blueberry pollination, where in red, our colonies more than often stayed where they were and were left in a holding yard. It seems like these blueberry colonies didn't recover quite as well going into October. That's where we're seeing our drop off in bees. But a lot of these blueberry colonies are sitting at around 6 frames of bees, or actually it looks like about seven, maybe 6.5 frames of bees, where our non-blueberries are about 2.2 frames of bees higher than that. And that continued over winter still, where our non-blueberry colonies still seem to be a little bit stronger by about 1.5 frames of bees than that blueberry counterparts. And again, when they're going up into blueberries, they're differentiating by about 2 frames of bees, or 1.95, with those non-blueberry goers being stronger. Interestingly, when they went into July, they really converged back to the same level. We had low levels of EFB infection that year, so maybe that could be why there wasn't a lot of damage from EFB in these colonies. And then down into October, we're seeing our colonies are doing uh, very poorly without proper management of a lot of diseases in EFB um, across this intensive study and intensive transport. So this is frames of open brood. We're starting very tightly close with the frames of open brood at about 1 frame of total open brood in February at the end of almonds. They seem to converge or diverge a little bit, about a 1/4 frame of brood difference here with that non-blueberry goers still a little bit ahead before they come back together to be about the same in July, and then crashing down in October to almost no brood for that overwintering period together. Coming out of winter into almonds, got a little bit of separation again, 0.21 frames of open brood difference between non-blueberry and blueberry goers. And then into May, they really took off. It seems like those non-blueberry colonies were able to put on a lot more larval growth, larval production compared to our non-blueberries, about zero point 0.8 frames difference there before converging again in July and into October. And then finally, here's our frames of capped brood. We have, let's see, we'll go ahead. It seems like for the first three, those error bars are overlapping, so there's not a lot of difference. Maybe a little bit, maybe a 1/4 frame of bees difference in that May before they come up to about the same, and then back down to 0. And then in that second year, we actually are seeing a little bit of difference in that capped brood, about zero point 0.4 frames difference in February, expanding up to that 0.74 frames of capped difference. So throughout this experiment, it seemed like our non-blueberry goers were had a little bit higher levels of bees, open brood, and capped brood than our blueberry goers. But again, we have that confounding variable that maybe it's the movement of the colonies into the blueberry that caused that stress and that lack of um, bee production. All right, so now instead of splitting them into uh, blueberry goers versus non-blueberry goers, we've got them split into infected versus non-infected. Where infected is orange, and this sort of gray is not infected. This is regardless of what treatment they went to, blueberries versus non-blueberries, and instead is again getting at the question of how impactful is a foul brood infection on colonies. We see a large difference, and in this case, they're starting out at a pretty different point in May in terms of frames of bees. So I calculated the increase in frames of bees as they're hitting their peak of population into July and August. And we see that those colonies that were not infected were able to put on quite a bit more frames of bees than those that had suffered an EFB infection at any point throughout the experiment. So it seems like EFB can take off a couple frames of bees in that summer uh, growth period in these relatively smaller hives. And so that could be a critical point. It's showing that EFB can damage your bee populations and weaken that colony. We're seeing the same pattern for open brood. And again, this disease kills your open brood, where moving from May into July with that big brood production increase, we're seeing about uh, a half of a frame of open brood more being produced in May to July, as opposed to the infected colonies that just had very, very little growth, actually. It seems like they were very, very stunted in their larval growth, maybe because those larvae are dying out to that EFB. And then again, with our frames of capped brood, this is still, some of those larvae that are dying, they're not making it to that capped brood season or capped brood stage. But it seems strange that we actually had higher levels of growth for our not infected colonies, and that's something that we'll have to tease out a little bit more. Or no, sorry, that is correct. There's higher levels of not infected frames, about 1.08 frames of not infected, for not infected bees, and then zero point 0.6 frames. That's what we expected to see. All right, I think I've got a couple more minutes and I'll leave some time for questions. But so the conclusion that we can pull out from what we've done so far and what we've analyzed so far from the study, it doesn't seem like there's a lot of evidence that blueberry pollination increases EFB symptoms in Washington. And it's a 2-year study. We're going to see much more data from Oregon State and the other universities come out. So far in Washington, we can't really say that blueberry pollination increased EFB symptom um, prevalence. But it does seem like there is some evidence that blueberry pollination is a little bit harsher on colonies in terms of amount of bees and brood than colonies that are being left at a holding yard. So if you move your colonies to blueberries, you really need to keep them fortified with additional syrup and pollen patties, which is something that we only fed syrup and not pollen patties this year, or those 2 years. So there may be some deleterious effects rather than leaving them in their place. Oh, and I should also say, then EFB, of course, is harmful to your colonies and will reduce the number of bees and brood and can result in early death of your colonies. So more to come. Again, we're going to have about double the inspection data at Oregon State University. They were able to keep in touch with their beekeepers very, very closely, and were able to track more of those colonies throughout all 8 inspection points, which was critical for this study. We're going to get the vitellogenin analysis, so how much of that protein is present in the bees' heads of the adult bees, to see how well they're being fed, how much EFB impacts that ability to store proteins in the head and then produce the brood food for larvae. We'll also see a laboratory analysis of bacterial presence, which is ongoing now and moving forward quite quickly, thanks to the folks at Oregon State University. And then also out of Oregon State University, we'll learn more about what bacterial strains of EFB are present in these larvae at different periods of time, and in the adults. We already know from Peter Fowler in 2025, that same study, that there's a lot of different strains of bacteria or of EFB bacteria, and they're split up into typical versus atypical groups based on their genetics. And typically, those typical ones are really not able to survive in oxygenated environments, and those atypical ones seem to be associated with a little more ability to grow in oxygenated environments. It seems like the atypical ones from their work has a little more virulence and co-infected colonies, so colonies that have both typical and atypical strains, they found have significantly higher rates of that clinical, meaning symptomatic disease levels. So I'm really excited to see what data we can get for our bacterial strains and compare it to that group out of Michigan State. And then eventually, coming in probably 2027, or we'll formulate or have evaluations of management techniques for EFB control. And then in 2027, we'll ultimately develop effective EFB management practices that we can distribute to keep EFB controlled and our colonies healthier. All right, that's all I've got. I think we'll move on into questions.
Awesome. Thank you, Ryan. That was so interesting. We do have a couple questions already. Um, how were the colonies in this study overwintered? Outside or in wintering sheds or indoor?
Great question. All of our colonies were wintered outdoors, so they experienced the harshest, most simple management tactic for colonies.
Were the colonies treated for varroa and with what and when?
Yep, we used a rotation of varroamiticides. They were treated in almonds. I think year one it was treated with oxalic acid dribble, and then year two was definitely treated with thymol in those almonds. And we also treated for them in late spring, just before they went on into honey production, and that typically was oxalic acid dribble. Um, I think year one it was oxalic acid dribble, and year two um, may have been thymol. That may have been switched. And then we treated them again in October, late October, again with I think usually oxalic acid. That tends to be our go-to is the oxalic acid dribble.
Okay. Is there any relationship between PMS or parasitic mite syndrome and EFB? Are they related through poor nutrition? Is PMS an identifiable disease?
Good questions. I do not know of any links between PMS and EFB. They're often confused for one another because the symptoms look so similar. Um, we still think PMS is largely about the buildup of viral diseases in these colonies and the weakening of the bees via those viruses, as opposed to EFB, which is the bacterial disease. But if you have a weakened colony from one or the other, from varroa mite and its viruses or European foulbrood, weak colonies are always more susceptible to other stressors and other diseases. So I don't know of a direct link, but I would not be surprised if there would be one via that route. Um, and then what was the you remember what was the second part?
Is PMS an identifiable disease?
PMS is sort of a complex. Identifiable explicitly, not really. Um, it's sort of an amalgamation of symptoms of this colony has a lot of mites and a lot of different viral diseases that are causing the colony collapse. So, um, it's kind of a catch-all term for we have too many mites and the colony is collapsing because of that.
What type of smell is EFB typically associated with, if you can describe it?
Yeah, to me it smells like sour urine. Um, sort of a sickly sweet, putrid, sort of sour urine odor. An American Foulbrood is really dead fish to me.
What statistical analysis was done on the data?
Yeah, good question. We're going to finalize that analysis, but we're going to end up doing linear models on them that can capture both the beekeeper, the random effects of beekeeper, location, all of those random effects in addition to it before we publish this data and finalize it.
Are there any data sets about weather conditions during the peak infection periods?
Hmm. We did not capture that. That is something we could go back and look at from sort of a course level, looking at past weather in each area. Um, in my knowledge of the literature, I don't know of any great um, studies that have compiled that, but I wouldn't be surprised to find some.
Okay, this is the last one so far. If anyone else has any questions, go ahead and start typing them in. So the last one we have here is of all the hives that were dead outs, what percent do you think is attributed directly or indirectly to EFB?
Good question. Slide back actually. I could give a little bit better answer there. So, um, we had about 92 and 96. We had about 188 colonies that we were able to survey for all 8 time periods. Of that, it seems like about 60 of them survived. And here I would
Wow.
guess it's probably 15 over 60, so maybe uh or no, it wouldn't be 15 over 60. It would be 15 over 188 minus 60. So maybe 11%. I'd say about 12%.
Very interesting. Okay, we got one more. Do you think migrating the hives to the almond orchards increases the sharing of disease over a larger area, i.e., hives coming from many different locations?
Yes. I would say yes, it's quite likely. Um if you have robbing behavior, those bees might be entering colonies and pulling out infected bacteria or bacterial infected bee bread and nectar and honey. So I think that it could increase that risk. And that is one of the major risks of moving all of our eggs into one basket each year in almond pollination.
After pollination in blueberries, were the hives fed and managed or just left to recover on their own?
Yep, we never did protein patties, but we did feed sugar syrup regularly. So we were monitoring the weight of those colonies and giving them sugar syrup. They would all receive sugar syrup if it seemed like some of them needed it. And so in that rate, they were given that syrup, but not maybe some of the pollen supplements that maybe they needed.
Do you know of any supplements that help strengthen the larval gut?
That's a good question. There's a lot in the works, I know. Um, I don't know of any that are proven to work at this point. I think there's a couple on the market, but as far as experimentally provable supplements, I don't know of any, but not to say that there aren't any.
Okay, awesome. Well, thank you so much, Ryan. It doesn't look like we have any more questions. If you think of any questions later, you're welcome to email Ryan. It's ryan.kuesel@wsu.edu. I will also send a follow-up email with the YouTube link once it's posted, and I can copy Ryan in there so you'll have his email directly. Thank you all so much for attending this first webinar. As a reminder, before you close your browser, you'll be prompted for that short 5 question education and outreach impact survey. Again, this is the first of 4 webinars this year, so tune into our other ones coming up. Our next one is in March. And again, this is recorded and they will be posted on our YouTube channel. So share it with your friends and rewatch it as you please. Thank you so much again for attending, and I'm going to end the webinar now. Thank
Thanks,
you all.
everyone. Thanks, Bree.